Hey, Miss Ray here. We're going to talk about the history of DNA. So getting into some exciting stuff. So some quick facts that you'll need to know is that chromosomes are made up of DNA and proteins. So chromosomes are found in the nucleus of the cell, of plant, animal, fungi, and protist cells. Chromosomes are made of DNA tightly coiled around proteins. Segments of DNA are called genes, and these special segments, the genes, are going to code for a specific protein that will then cause a trait. So an example of that would be you have a gene in your DNA, so there's a segment of DNA that is going to code for a protein that makes your eyes the color that they are. So chromosomes made up of DNA and proteins. A segment or a small piece of that DNA is called a gene, and genes code for specific traits. So first we're going to talk about the history. So we're going to take ourselves way back into time, back in the 1950s, before the 1950s, when scientists weren't sure what the genetic material was. So today we know genetic material is DNA. It's pretty common knowledge. We know that DNA and RNA is responsible for heredity. But back then in the 1950s and prior to the 1950s, they didn't know. They knew that there was something responsible for carrying the genetic code. They knew that it was responsible for heredity, which is passing of traits from parents to offspring. But they didn't know if it was DNA or protein. So we're going to talk about the three scientists who worked to first find the question about what is the genetic material, and then also solve the answer to what is the genetic material. So please, 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 as we go through these experiments, do not try to write everything down. I do have a summary slide for you to write in your notes. So don't try to write all of this in your notes, only that summary slide. So I'm going to talk through it a little bit. So Griffith, he actually came across this question on accident. He was looking to try to find solutions for pneumonia in soldiers. And he used two types of bacteria. He used pathogenic bacteria, which make people sick. It causes disease. And he also used non-pathogenic bacteria, which is a bacteria that doesn't make you sick. So in the side on your notes there, you might want to make a little star and say pathogenic equals makes you sick. So here's what he did. First, he did a control. He took a pathogenic bacteria he knows is going to make the mice sick, injected it in the mouse died. He knew that was going to happen. That was a control. He did another control in which he took a live, non-pathogenic bacteria, which doesn't cause disease, injected it in the mouse, and the mouse lived. Yay, he knew that was going to happen. It was a control. He knew those bacteria wouldn't make the mice sick. Then he killed the pathogenic bacteria, which would normally make a mouse sick. So he killed it, he injected it in the mice, and the mice lived because he killed the bacteria first. But over here with experiment D, something interesting happened. He took the killed pathogenic bacteria, which don't kill mice, they lived when it went alone, and then when injected alone, non-pathogenic bacteria makes the mice live, but when he mixed the killed pathogenic bacteria with the live non-pathogenic bacteria, again alone, are harmless, when he mixed them together, the mouse died. So alone, they're harmless, but when you mix them together, it can lead to disease and death. So this raised the question of, there had to be something still around in that pathogenic bacteria, even though it was dead, there had to still be something in there that was transforming the live bacteria, something that was causing disease, something moved from the bad dead one into the living good one, and it made the good one bad. And they called this the transforming factor, something that moved from the dead bad bacteria into the living good one. And it's something that caused disease that made the mice die. And they call that the transforming factor, which was going to be the genetic material. But again, they didn't know if that genetic material was DNA or protein. But this experiment raised the initial question. So here's what you're going to want to write in your notes. It's just a summary of what we talked about. So Griffith noticed that killed pathogenic bacteria, when mixed with non-pathogenic bacteria made mice die. And again, that was shocking because alone, both of them are harmless. But when you mix them together, the mice died. And he noted that there had to be something that was transmitting disease. And they called that thing the transformation factor, the transforming factor. And it raised the question about what is the transforming factor? What is the genetic material? And again, they didn't know if it was DNA or protein, but it raised the question. So you might want to pause, make sure you jot this slide down in your notes. So after Griffith's experiment, these next three scientists came along to answer the question he came up with. So the Avery, McCarty, and McLeod experiment tried to answer that question. What is the transforming factor? What is the genetic material? And they did two quick experiments. The first experiment was pretty easy. They injected protein into a bacteria and it didn't do anything. But when they injected DNA into the bacteria, they saw transformation. They saw something happen. And then in experiment 2, what they did is they destroyed all the proteins. When the proteins were gone, transformation still happened when the proteins were gone. So they knew the proteins weren't that important. But when they destroyed all the DNA, transformation stopped. So in summary, this is what you're going to want to jot down in your notes, is that they noted that when DNA is present, transformation takes place. And what that means for them is that DNA could be the genetic material. It could be that transforming factor. So their experiment showed DNA is possibly the transforming factor. But in science, you got to show it at least twice to prove that it's true. So this brought around our next group of scientists. And their names are Hershey and Chase. So Martha Chase and Alfred Hershey did an experiment. Again, do not write all of this down. I'll summarize it, and then there will be a slide at the end where you can copy. So Hershey and Chase worked with bacteriophages, which are simply viruses that infect bacteria. So they worked with viruses that infect bacteria. So the key parts of this is what you're going to need to know is that a virus looks kind of like this guy here. They have two parts. They're made up of DNA on the inside and protein on the outside. So DNA on the inside and protein on the outside. And they used radioactivity to label the protein and to label the DNA and see which one was transmitted. Because a virus makes something sick by injecting its genetic material into it. So what they decided to do is if I make the protein radioactive and then the organism that gets sick, the bacteria that gets sick, if it's radioactive, then I know that the protein moved, and protein is the genetic material. However, they knew if they labeled the DNA and made the DNA radioactive, if the DNA is the genetic material, then the DNA will be passed to the bacteria, and the bacteria will become radioactive. So I'm just going to go through my little summary slide here. This is my little drawing, and this is what I recommend you guys drawing in your notes. So this is a virus, and again, virus is made up of two parts, protein on the outside, DNA on the inside. The viruses that we're working with are going to infect bacteria, so my little blob here represents a bacteria. And if something is red, that means it's radioactive. So what they're trying to do is figure out what is the genetic material by figuring out what is passed. So in experiment 1, they made the protein or the outside of the virus radioactive. They let it inject its genetic material into a bacteria, and in the end they realized the bacteria was not radioactive. Because it passed on its DNA. But then to confirm that DNA was passed on, in experiment 2, they made the DNA radioactive. Then they let the virus infect the bacteria, and the bacteria became radioactive because the radioactive DNA was passed on. So what this means is that DNA is transferred from the virus to the bacteria, meaning DNA is the genetic material. So Hershey and Chase were the ones to finally put into the history books that DNA is the genetic material. So you want to pause, make sure you get this down. If you don't understand it, please come to office hours. It's a complicated experiment to do in a couple minutes. So this is going to bring us to the structure of a DNA molecule. And again, this brings us to a few more scientists. We have Rosalind Franklin and Maurice Wilkins. So Rosalind Franklin took X-rays of DNA and created photographs. So she took X-rays of DNA to make photographs, and those photographs were then used to determine the structure or the shape of DNA. So here is what a X-ray of DNA looks like. You'll want to be familiar with this image. Sometimes it will pop up on your SOL. So if you did not know, DNA stands for deoxyribonucleic acid. So ribose describes the sugar. So deoxyribose is the sugar in DNA. So deoxyribo is the sugar, and it is a nucleic acid. So deoxyribonucleic acid is DNA. DNA is double-stranded, and it's going to determine the traits of an organism. So DNA codes for specific proteins that then give organisms their traits. So a quick review of nucleotides. Remember, way back in biochemistry, we said DNA are made of nucleotides. DNA and RNA are made of nucleotides. And a nucleotide contains three different parts. It contains a sugar, which is going to be the pentagon here. It contains a phosphate group. Whoop, I didn't mean to do that, which is the yellow part here. And it contains a nitrogen base, which is represented by, that's either peachy or orange here. And these phosphates are in the wrong spot, so I'm going to kind of move them around here a little bit. Phosphates are on the side. Kind of like that. So nucleotides are made up of three parts: the sugar, which is a pentagon, phosphate, which is the yellow circle, and a base on the side. So here is a nucleotide for you to sketch out and label in your notes. Again, you have your sugar. In DNA, the sugar is called deoxyribose. Again, that's where DNA gets its name, from the sugar. It has a phosphate on the side. Phosphates are typically represented with a circle. And then a nitrogen base. And then the nitrogen bases have different structures we're going to talk about here in a few minutes. So our next two scientists, Watson and Crick, they are credited for finding the structure of DNA. So Watson and Crick discovered that DNA is made up of two chains of nucleotides joined together by nitrogen bases. And together they form the double helix. And you might be wondering why I said discovered like that. And it's because they stole the work of Rosalind Franklin to help come up with their discovery. And she didn't get credit for her work until years later when they found out. But Watson and Crick, they got a Nobel Prize. But again, if they didn't have that work from Rosalind Franklin, they wouldn't have made their discoveries. So they discovered that DNA is two chains of nucleotides. So there's two chains of nucleotides in a DNA molecule. They are bound in the middle with hydrogen bonds. And they twist together to form a double helix. So double helix is the structure of a DNA molecule. It kind of looks like a spiral staircase. So again, there's two strands held together with hydrogen bonds in the middle. And then they twist together to form the double helix. So here is another look at the nucleotide. You can see the deoxyribose sugar, the phosphate, and the nitrogen base, all labeled. Note that the nitrogen base does contain carbon rings, and we're going to talk about the two types of bases here in a few minutes. So the backbone of DNA is made up of alternating phosphates and sugars. So the backbone, or the sides, the rungs of DNA are made up of sugars, deoxyribose sugars, and phosphates. So the backbone is often called the sugar phosphate backbone because the sides are made up of sugar and phosphate. And then in the middle, you're going to find your bases, which are like the rungs of the ladder. They're located in the middle. So the sides are the sugar and the phosphate, and in the middle, you find your bases. And your bases are what's going to be linked through hydrogen bonds. So if I were to draw the hydrogen bonds between, so this is one side of the DNA molecule, whoop. Here's the other side of the DNA molecule. Note that in order for these to line up, one side is right side up, one side is upside down. So one side right side up, one side upside down. That is called anti-parallel. They're called anti-parallel because one side's upside down. And if I were to draw in the hydrogen bonds that connect the two sides of the DNA, I would draw dotted lines. Again, hydrogen bonds are represented with dotted lines between the bases. So I would draw dot, dash, 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 dash between the bases here and between the bases here. The number of hydrogen bonds between the bases depend on which bases are being linked. In a few minutes, we're going to talk about the different bases. Essentially, an A and a T have 2 hydrogen bonds, so 2 dotted lines, and a G and a C have 3 dotted lines, 3 hydrogen bonds. So like I said, there are 4 nitrogen bases. We have adenine, which is represented with an A, thymine represented with a T, cytosine represented with a C, and guanine represented with a G. Typically, we're just going to say these as A, T, C, and G. Those 4 bases are divided into 2 groups. We have the pyrimidines. Pyrimidines, really long word, but a small structure. So really long word, but only 1 ring. And the way you can remember these is because pyrimidines, both of the pyrimidine bases have Ys. Thymine and cytosine both have Ys, and so does pyrimidine. Long word, small structure. So thymine, cytosine are pyrimidines because they have Ys, and long word means small, one single structure, 1 ring. Purines, small name, but larger structure, because purines have 2 rings. So purine bases have 2 rings, and our purine bases are the only 2 that are left, which are adenine and guanine. So when you have your 4 listed, just take your ones with a Y and add them to the pyrimidines. Those have 1 ring. The 2 that are left are your purines, and they have 2 rings. And then we're going to talk a little bit about how our bases pair. So we talked about our 4 bases, A, T, G, and C, or adenine, thymine, guanine, and cytosine. And they always pair a certain way. A always pairs with T. So adenine and thymine go together. While cytosine and guanine, or C and G, they always pair together. The way that I remember this is because C and G, they both are curved. The letters are both curved, so I remember that's how they go together. And then A and T, they're kind of straight lines that make those letters, so they go together. And these are called complementary base pairs because they always go together. A and T are complementary, they pair together. And G and C are complementary, and they pair together. And as you can see here in the pairs, A and T have 2 hydrogen bonds, whereas G and C have 3 hydrogen bonds. So the way that they pair is called Chargaff's rule, which essentially means A, the number of adenines, equals the number of thymines, because since adenine and thymine pair together, you're always going to have the same amount of adenines as you do of thymines because they're paired together. So A goes with T, and then the number of guanines equals the number of cytosines because they always pair together. So you're not going to have a different number of guanine and cytosine. You can't have a different number because they always pair together. So they're going to be the same number. So A equals T, G equals C. And that is called Chargaff's rule. He's often our forgotten scientist, so try to put him to memory there. Chargaff's rule, A equals T, G equals C. If the curved letters go together, don't quite make sense to you, you can also remember it this way. Apple trees, A and T, and collard greens, C and G. Just a little mnemonic device. I remember curved ones go together, but again, apple trees, A and T, collard greens, C and G. You can remember it that way too. So here we have a little practice problem. I'm going to read it here. I'll do the math on the next slide. So the first one, if one strand of DNA contained the sequence A, T, C, G, G, A, T, what would we find on the complementary strand? So this is going to be doing our base pairing rule. So to figure out the opposite strand, I'm essentially going to say what goes with A. T. So A goes with T, so I'm going to write a T. T goes with A, so I'm going to write an A. C goes with G, so I'm going to write a G. G goes with C. G goes with C. A goes with T. And T goes with A. So to find the complementary strand, you just match them to who they pair with. And then our second question, I'm going to do the math on the next slide. If a segment of DNA contains 100 nucleotides, so I have 100 total, 30 are A, how many are there of each of the other bases? So the way that I would figure this out, it told me I have 30 A's. Chargaff's rule states A equals T. So if I know A is 30, then I also know that T is 30 because A equals T. And I have 100 total. So it told me that I have 100 total. So I'm going to take away my 60. This is 60 total. So out of my 100, 100 minus 60 gives me 40. So I have 40 left. 40 of my 100. And from there, I know that the number of C's equals the number of G's. I know that these two numbers have to be the same. So what I'm going to do is take my number that I have left and divide by 2. So I have 40 left, and if I divide by 2, 40 divided by 2 equals 20. So I know that both of those are 20. And our nucleotide sequence, or our sequence of bases that we have in DNA, these are very, very important because these are going to be what causes and contributes to our genes. Our specific bases and the order that they fall in make up our genes, and our genes make our proteins, and our proteins give us traits. So these are going to be very, very important in determining the traits of an organism. So if you have any questions about this, please visit your teacher's office hours, and